Thank you very much for the kind introduction and for this opportunity to be able to talk to this amazing audience, both present here as well as the many thousands I hear that are going to be listening in through the remote means. So I'm going to begin with a slide that really brought me to immunology. And what this epitomizes is both the complexity as well as the simplicity of the system. So what I have shown here are the many cell types, pathways, and processes involved in mounting an immune response. And as you probably realize, this is not even half of what people know today. But if you boil it down to the essence of what the immune system does, for my simple mind, I look at it in terms of three functions and two questions. And the three functions the immune system does for us, for our pets, is one, to keep the bad stuff out, that's the barrier function. To check if something has gotten through, that's the surveillance function. And then to deal with the intruder, that is the defense function. And it does by asking two very fundamental questions. Is this me? And if the answer is no, what am I going to do about it? What's my next response to be able to protect the organism from these challenges? With that perspective, what I want to talk today is to discuss and answer a couple of questions. One, why enhance the immune health of an otherwise healthy individual or animal? I'm going to be focusing most on the companion animal side of things. We had some amazing lectures that gives the overview from the human perspective. The second, how can we use food to influence the immune system? Having trained as an immunologist, never in my remotest thinking I would have imagined that food could have had an impact, and lo and behold, here I am talking about nutrition immunology. So I'll walk you through some of those thinkings and challenges that I've worked through in my own mind as we evolve some of the research that we have done, and then present to you a framework how to understand or consider the interaction between nutrition and the immune system. So let's begin by dissecting the first question. What are the reasons you need to impact or enhance the immune status of an otherwise healthy dog or cat, equally applicable to humans as well? So what I have here is a diagrammatic representation of immune strength or the ability to fight infection at various life stages of a dog: puppy, middle-aged, and senior. And what you can see is both at puppyhood and in an older animal, your ability to fight infection tends to go down a little lower, and we saw great examples and discussions of that. Middle-aged dogs, adult dogs, typically tend to have a healthy immune response, and this is true for cats as well as humans. Again, we're not going to be talking about other causes where immunosuppression can happen. This could be fatigue. There are several studies that show that extreme athletes do undergo a phase post-exercise where they are exposed and suppressed from an immune perspective, and therefore are very susceptible to various infections, respiratory infections, for example. Another, of course, is disease. So that's one. Life stage can have an impact on the immune system. Second is underweight and overweight. And we got some great presentations, so I'm not going to belabor the point. Clearly, protein energy under nutrition can lead to various immune defects as well as infection, and so is obesity. And if you dwell down into some of the mechanisms, and again, I'm not going to be able to go into all the details, I've just captured certain pertinent points for us to think about, and we can discuss more. Clearly, a lot of immune response depends on a very good source of protein to be able to make these antibodies and cytokines that drive the immune response. And then on the obesity side, things like leptin that are being produced, other cytokines that are produced by fat cells, tend to lead to low-grade inflammation and a phenomena called as immunosenescence, and we'll talk a little bit about that. There is data showing that non-esterified fatty acids can also activate toll receptors. We saw a great presentation on that. That can also enhance immune inflammation, low-grade inflammation in pets and humans. Then if you talk about life stage, we saw examples in puppies. There have been studies showing that the immune response measured by proliferative ability can be quite significant as compared to the adult. Secondly, the immune response in a young animal, as in a young human, tends to have a Th2 bias. And if you start looking at the reasons behind that, one of them seems to be inefficient antigen presentation, which is the key step for mounting an immune response. There is also the continued presence of immunosuppressant factors post-birth. And then there is a unique phenomenon that's been described in young neonatal cells, where when you have a Th1 response, because of the absence of the appropriate dendritic cell support, you tend to have apoptosis rather than proliferation. And that is because of a unique combination of receptors that are expressed in these neonatal Th1 cells, which is a combination of IL-13 and IL-4. Lack of dendritic cells lead to production of IL-4, and when IL-4 binds these receptors, you have apoptosis instead of proliferation. If you look at the senior dogs or older animals, you tend to have also a very similar phenomena, but for different reasons. Again, suppressed immune response. There is obviously a declining in both the hematopoietic bone marrow as well as thymic atrophy. There is increased oxidative stress, low-grade inflammation, and this phenomenon of immunosenescence, where immune response is not as effective, as immediate as you would like it to be, as seen in younger animals. And then there is the effect of metabolic stress, low-grade inflammation, resulting in another phenomenon that's getting a lot of attention in the human space called as inflammaging. That leads to decline in immunoplasticity, reduced responses, and also supports or leads to immunosenescence. And again, if you look at the reasons behind that, inefficient antigen presentation, cellular changes, as well as apoptotic increases in the cell responses, and decrease of an important cell type called regulatory T cells. Now, going on to another thing that can also impact the immune system or immune response is stress. And stress is a very common phenomenon that we all talk about. Both our younger twins who went through college applications, so me and my wife and our older son have been stressed since about December, right? And now we're getting to open those applications, seeing where they end up. So stress is a common phenomenon in all our lives. But stress has a very important effect on the immune response. In fact, there's an elegant study that was done by Glasser et al., where they did two different studies. One was studying kids in medical schools, students in the medical schools, two groups, one that had a stressor in their life and a stressor were preparing for board exams. And I'm sure many of you have had that experience. I didn't go to med school, so I haven't seen that, but I've heard it's extremely stressful because that sets you up for your residency and the next level of education. And the other group did not have those stressors. When you immunize both groups with the same vaccine, you got a significantly lowered response in the group that was stressed. Now again, these are young people, no issues with their immune system. Another study was done with older adults, and in this case, one of them was taking care of a significant other who had challenges, health challenges. And the other cohort had couples that did not have that health challenge in their lives. And when you again compare the responses, you see exactly the same phenomena. Stress suppresses immune response irrespective of your age. And if you look into the mechanisms, several have been noted, all revolved around phagocyte function. There are immunosuppressive factors or roles that played by stress hormones like glucocorticoids, epinephrine, norepinephrine, as well as cortisol. So several conditions that we saw life stage, obesity, malnutrition, stress can lead to significant decline in the immune response or the ability to respond effectively to an immune challenge in an otherwise healthy animal. And again, for my simple mind, I like to go back to high school graphics and look at Venn diagrams. And one of the things that pops out as you look at various pathways, when you start thinking about what are the elements that you can engage with to be able to support or address this condition, two of them stand out. One is macrophage function, and the other one, the role of regulatory T cells. And again, by far, this is not an exhaustive list, but this is how we would approach the problem in trying to address and look for a solution. So quick recap, we already went through that, so I'm going to go through past that for the sake of time. So after kind of work through the first question, let's ask the second question: how can food influence the immune system? I have the good fortune of speaking after some excellent lectures who covered that, but let me make an attempt to kind of give my own take on that. We went through these slides, let me go back, yeah. Dr. Joseph covered a lot of this. Largest immune organ, 65% of the immune cells are present in the gut, and 90% of the IgG producing cells typically reside in the gut. Again, this was something that I learned after I kind of changed over to the nutrition immunology space, because if you'd asked me earlier, I would have responded spleen. That's not the case. Unique anatomical features, again, I'm not going to belabor the point. Huge exposure to antigens as part of the 2,400 kilograms of food that you typically eat on a daily, during your lifetime in an adult human. And millions or different species of bacteria that you're exposed to. And again, we saw diagrammatic representation of that. But the fundamental question that I would propose, or I present to you as an argument, is that what we eat, what we feed our pets, interacts with a significant part of the immune system. And when I sometimes go to high schools and talk to students, it's interesting to be able to reflect that what I had for breakfast this morning is actually talking to my immune system right now. So if you look at it from that perspective, there's an opportunity for us to be able to engage with the immune system in meaningful ways to influence in a direction that might be effective in addressing the overall health. Now, how do you do that? That's the million-dollar question. And again, I'll propose some ideas for us to look into. So we again saw great examples of individual nutrients, some phytochemicals that Dr. Maidani talked about, arginine, great lecture on that. But again, I've pulled out a few examples of some of the key elements that are present in what I call as phytonutrients, or why your grandma would ask you to eat broccoli, which is good for you, right? So let me just list out all the references. So the gut-associated lymphatic tissue initiates T-regulatory responses to food antigens, and that's why if you take a microgram of that food and you put it into your blood, you would have a septic reaction in some cases, but you eat all the hamburger and cheese and other things, nothing happens. It's because you have an effective Treg function in the gut. And this is a process that's driven by CD103 positive dendritic cells that lead to increase of T regulatory cells using the cytokine TGF-beta, as well as a metabolite that is produced from vitamin E. AHR, aryl hydrocarbon receptors, induce expression of FOX3+, which are important T regulatory generators. These are again present in several cruciferous vegetables and are key bioactives, perhaps some of the components that's present in vegetable and fruit diets that were described earlier. And there are other factors such as NRF2, nuclear factor erythroid 2 related factors, that regulate key antioxidants such as glutamine using NF-kappa B pathway. So these are some of the elements that's present in diets, and there are many more. I'm just talking about a subset. And there's ways for us to be able to see how we can incorporate those as part of the diet to be able to perhaps communicate and talk to the immune system and direct it in a path where we want it to go. I want to spend a little bit of time to really talk about this triad. You have the diet, you have microbiota, and you have the immune system. The directions of the arrows are significant because the black arrows talks about what's being delivered from one end to the other, and then the white arrows talks about what gets back. So for example, diet provides vitamin, fat, iron, sugar, and fiber to the microbiota as well as to the immune system. And then microbiota ferments that we heard about that in terms of returning back to the nutrient, to the dietary component that you feed a pet or a human. Again, microbiota contribute metabolites such as short-chain fatty acid, as well as several nutrients that enhance and interact with the immune system coming from the microbiota. And then immune response plays a role in trying to keep some of that bacteria at bay. We talked about IgA being produced, other antimicrobial factors that are released in the gut to be able to see that none of that gets into the systemic circulation. And then, of course, you have nutrients that are present in diets, such as vitamins. We heard a lot about that. Protein, iron, magnesium, selenium, zinc, toll receptor agonists, other ligands from diet that can interact with the immune system receptors to be able to activate that. So again, a quick recap. Again, going back to two questions: malnutrition, life stage, overnutrition, stress can lower immune status. Inefficient macrophage and regulatory T functions are two things that I like to focus on, but there's others, obviously, that could play a role as well. Gut's the largest immune organ. We saw that over and over again. And then functional foods can stimulate the gut-associated lymphoid tissue and likely restore optimal function. So let's now dive into understanding or perceiving how nutrition can impact the immune system. And here's a proposed framework that I'd like to walk you through. I've broken down into 4 stages. Stage 1 is complete and balanced nutrition. Now, in many situations that we typically interact with, you're talking about, at least in the pet situation, most of our diets are complete balanced, whether it's ours or any of the other products out of the market. So it's not a big concern. There could be some issues with micronutrient deficiency, but it is important to remember that there are situations out in the world where malnutrition is real, can have a significant impact on the health, in particular immune health, and therefore exposure to infections, etc. So that is something to keep in mind. And we saw some great examples of micronutrients today as well. Stage 2 is you're trying to optimize both micro and macronutrients. We talked about vitamins, we talked about minerals, we talked about specific components of amino acids. One way to look at all these is that these are passive ways of engaging with the immune system. And that's a point that could be argued in either way. Passive in the sense that it gives things to the immune system for it to be able to do its function. Now, I'm not a golfer, but imagine if you give me all the golf gear and you put me on a green, I'm not going to be able to get the ball very far. That's because just because I have the gear doesn't enable me to do the job. So that's what I mean by passive versus active. You need to be able to actively engage. And these are important. Don't get me wrong, that's probably the first step. But active takes it to the next level in my view, and this is something that we can discuss and debate about. The third stage is where things get a little interesting, where you're talking about active engagement with the immune system. And this is where you look at some of the pathways that we talked about earlier and look at your toolkit in terms of nutrients, because you're purely talking about nutrition approaches, not anything else. And how can you come up with a set of ingredients that could likely impact some of these pathways and affect the immune system in ways that you want it to be modulated? And in some cases, you want to enhance it. In some cases, you may want to suppress it if there is local inflammation, for example. And the fourth is personalized nutrition. This is kind of the holy grail. A lot of work is happening in that space. I was fascinated to hear about the personalized nutrition from maternal antibodies coming in. But this is where you're talking about surgically addressing a specific problem that a given pet may have at a given time in their life. This is equally applicable to humans as well. So stage 1 is very clear, very simple, so I'm not going to belabor that point. Let's go into optimizing micro and macronutrients. Talking about high quality proteins, high level of vitamins, A, C, E, I should have probably added D as well based on some of the work that was described. Minerals. Calorie restriction strategies could either fall here or active. Again, it's debatable. This is not a perfect way of characterizing things. And then antioxidant supplementation, which counters the effect of oxidative stress. self-inflicted damage due to free radicals, and maybe even help in this process of reducing inflammation and perhaps correct immunosenescence. Stage 3, you get into a very interesting situation where you're looking at ingredients that you can add in diet to be able to address specific pathways or engage with specific pathways to enhance your immune response. And I've listed several. Many of them come from the microbial immunomodulator class, beta-glucans, mannins, microbial nucleic acids, and some proteins. And this is where probiotics, a lot of those things would fall in. There could be also immunomodulatory ingredients. We've done some work with bovine colostrum that I'll talk about. Whey proteins have been looked at. We also did some work with spirulina that I'm not going to be covered for the sake of time, that have very interesting applications in terms of enhancing immune status both in young as well as adult animals. Managing inflammation using fatty acids like DHA and omega-3 to be able to suppress that could be another ways to look at. And then dietary components. We're looking at things that can target the ARH pathway and NRF2 pathway are things that we can look at in this bucket. Short-chain fatty acid is something that's very near and dear to many of our hearts because of the work that we do with microbiome and the gut. Those are very important because they drive the production and the generation of an important class of cells called regulatory T cells, and they play a very important role in trying to suppress inflammation and perhaps manage some of the inflammation that happens in older pets. So that would also fall in this category. With that, I'm going to switch over and walk you through some studies that we've done with colostrum. A couple of things I want to kind of mention as we walk through those studies. These are studies that were done with dogs. There's a study with cat that I'll talk about. The question that always comes up is, how do you explain the mechanism? And that is something that we are not able to do because of the nature of studies that we do. We do not have no invasive work is done. Most of the blood samples that you see that are being collected as part of this were part of regular veterinary care. That's important for us in terms of how we approach our research. It's both a limitation, but I look at it as an opportunity. And this is where we come up with creative ways to be able to come up with tools that you can use to answer the same immune question, which is, does this make a difference for the pet? So the first study that I'll talk about is what I'd call as an active immunomodulation using bovine colostrum. Here's an outline of the study. This work conducted with Alaskan dogs, 24 dogs, 2 groups. Adults 2 to 6 years of age, 2 diets. There was a control diet and a test diet, which had colostrum supplementation. It was a 40-week trial that was conducted in Alaska, and we had both immune and gut health measures monitored during the study. And one thing that I've learned as I got into this field of so-called nutritional immunology is that nutrients are not drugs. You can expect to have an immediate effect with a TNF-alpha blocker. You can have an immediate immune response to LPS. Nutrients take a while to take their effect. And therefore, doing studies that give you enough time to be able to ask and answer the question whether it works or not is important. If you do short-term studies, 6-8 weeks, in many cases you really don't have enough time for the nutrient to be able to engage with the required pathways to initiate a response, and therefore you end up with questions that are that say things did not work, but in reality, if you had persisted a little longer, you would start having results. So most of the studies that we've done, now 44 is an empirical number, but 6 months and beyond is important for us to be able to demonstrate the efficacy of a nutrient that has immune enhancing abilities. So this is a 40-week trial. And I'll quickly walk you through the design of the study and some of the metrics that were used to measure immune response. The other important thing to keep in mind is that when you talk about adult dogs and cats, you want to be able to give a period for whatever diets they're on to be able to all come to the same baseline level. We've seen that in several studies that we conducted. So we typically give about 8 to 10 weeks of pre-test phase where all the subjects or the participants in the study tend to be on a control diet. And then we switch over to the test diet, which is what you see. You have 8 weeks of pre-test where both groups were given the control diet. And then at the end of the 8 weeks, test group was shifted to the test diet. The control group remained on the control diet. We use CDV vaccination in this particular study to be able to ask the question, is the immune response better in the group that had a supplemented diet? Now, as many of you know, there are other ways to be able to do that, but from our perspective, starting to take a look at it from the whole animal point of view and see, does this make a tangible difference in the immune health of the pet? The vaccine response is probably one of the most simplest and the most elegant way to do that. And again, something that we could discuss and argue about, but that's how we've done most of these studies. It's also a holistic measure of all the pathways that come into play to be able to initiate a response. So it's a great way for us to be able to so-called kick the tires of the immune system and see how it responds. The study, we had monthly samplings. Again, this coincided with their typical vet visit. That's important for us from how we do and design our studies. And here are some of the immune evaluations that we've done. We evaluate local immune response by measuring fecal immunoglobulin. There are several studies that show that fecal immunoglobulin is an important marker on how effective your gut immune system is. And in most studies that we have done and others have published, increased level of fecal immune response translates to a healthier immune system of the gut. Again, you're not talking about numbers that go through the roof, but overall increase, statistically significant increase compared to the control, is a great way for us to be able to measure and see how the local immune response in the gut is behaving. And then we talked about systemic immune response, looking at CDV vaccination. We also did some gut health measurements, but I'm not going to talk about that because of time. But again, for us, that's an important way to benchmark how the immune system enhancement impacts gut microflora response in the GI tract. And in most cases, what we see is that if the dogs or cats have some level of stress, these dogs fare much better if they're on a diet that is immune boosting. Another important measure that's not listed here, but we always cover, is to start looking at, is there any overt sign of inflammation? Now, it's a question that many clinicians sometimes are concerned with, is say, you're talking about an immune-enhancing diet. Does it lead to overt inflammation? And in most cases, the answer is no, but we have the data to show that. Because in my mind, when that question comes up, the question I go through is, you don't realize how hard we try to be able to enhance the immune response. To be able to get to a point where you have overt immune response across the board is very, very unlikely. But nevertheless, you have to have data to prove your point. So we'll talk about that in a bit. So let's look at immune response from the local immune system. And what we have here are fecal IgA responses. I just have 2 data points marked, but there were others in between. And what you could see is that in the control group shown in black here, the response were more or less stable, which is what you'd expect. There's going to be a bounce, but remains within that range. But you can clearly see an enhanced immune response in the group that was fed a diet that was supplemented with bovine colostrum. Now keep in mind, this is not canine colostrum. This is bovine colostrum given to an adult animal. So the mechanism of action is very different from absorption through the gut. It's most likely interacting with elements of the gut immune system, clearly demonstrating that you are having a local immune effect in terms of higher level of fecal IgA production. When you look at responses to canine distemper virus, we had to look at it a little differently because all these dogs were exposed to the vaccine before. So you're looking at responses over above their baseline. So that's what's plotted out here. And what you can see is that when the study was started in the July timeframe, you're looking at a time point, and then you start seeing the booster effect in both groups. What is very striking for me, this was one of the first studies that I was involved in, was this response stayed higher almost until the end of the study, clearly demonstrating that diets doing something to the immune system that is being manifest as overall immune boost, which is what the data represents. And this is the data to show that there is no overt immune stimulation. And what you're looking at is C-reactive protein, which, as all of you know from a clinical perspective, is a marker to look for acute inflammatory responses. And you can see that the numbers were almost comparable between the control and the colostrum-fed groups, and well below or well in the range of the normal CRP levels that you'd expect in these pets. So clearly the immune response, you can see markers of that both in terms of local activation in the gut, there is activation in the systemic immune response, but there is no overt stimulation in terms of overt immune response that could be a concern. So the key findings were in colostrum-fed diets or colostrum-supplemented diets enhance systemic immunity in terms of vaccine response that's stronger and lasts longer, enhance local immune response in terms of increased fecal IgA produced by the gut-associated lymphoid tissue. I did not show you the data, but these dogs were able to handle stress-related gut problems much better. And then we saw the data showing that there is no overt immune stimulation based on the CRP data. So clearly, higher levels of immune response and healthy animal. And this was published in the British Journal of Nutrition in 2013. Since this study, we have also done another study with adult dogs, and these are dogs in the senior groups that will be published shortly, so we will have data hopefully if there's another forum down the road, where colleagues of mine who did the work were able to also demonstrate increased level of T-regulatory T cells in these dogs. So that's fascinating work. And again, one of the things that I was telling to some of our colleagues working in the human space, one of the big learnings for me coming into the companion animal area is the limitation of resources we have in terms of reagents. Antibodies, cell types, very limited. So the team did an amazing job to be able to find antibodies that can cross-react, to be able to look for some of the Treg responses, and hopefully we'll share that in the future. The second study that I want to talk about is a study that was also done with colostrum button kittens. I don't want you to leave with the impression that I'm all dog-focused. There's cat studies that we do as well. Very similar study, but these were young kittens, 24 kittens, 8 weeks of age, post-weaning, 2 diets, control and test. And again, the same pattern. We want to be able to have these studies run long enough to be able to give the immune system to respond to whatever dietary interventions we make so that it can be reflected in the measures that we take. Very similar pattern again in terms of the parameters that were tested. We look for markers for local immune response, which is IgA. We look for markers for systemic immune response, and this we looked at for rabies vaccine response, which is where it's a different vaccine, again, asking the same question, are you able to respond better to a challenge? And then a different marker for looking at overall immune activation or inflammatory stress, which is serum amyloid protein A. There were measures to evaluate the effect of stress on the gut of these cats. Again, I'm not going to talk about that. We did some microflora profile changes, but most of this work was prior to some of the 16S data that was available, so it's more looking for pattern changes as well as some of the indices like Simpson indices that people talked about. Here's an overview of the study. Again, what you see here is a little shorter pre-test period because these were young animals to begin with. Rabies vaccine administered at the end of the pre-test phase where the test animal was shifted to a diet that was supplemented with colostrum, and you had monthly samplings taken. A booster vaccine that was administered at week 38 as per the protocol, and then you start measuring responses to these vaccines. So what you're seeing here is looking at the titers to rabies vaccine. And what you can see is that start measuring from week 36 onwards, you're seeing a significant bump in the antibody response or the titer response to the rabies vaccine. Again, the pattern here is a little different than what we saw with dogs, but these are different species, different vaccine responses. You'd expect a different behavior in terms of how those things trend. But clearly significantly higher levels of rabies antititers in the cats that were supplemented with diet supplement with colostrum. You could also see much of a varying trend, but again, towards the end, you start seeing the separation when you start measuring fecal IgA, which, as you all know, is a marker for activation of the gut immune system. Significantly higher levels in the cats that were, or the kittens that were supplemented with diets fed with bovine colostrum. And then the marker for establishing that there is no overt immune stimulation, we looked at serum amyloid A as a marker for, it's an acute phase protein as a marker for overt immune stimulation, and clearly numbers are well within the normal range, showing that while you're seeing significant immune enhancement both locally as well as systemically, there is no overt change in the overall inflammatory status of the pet. Summary again, colostrum increasing systemic immune as well as local immune response. We had some data on the gut behavior, and again, we saw the same pattern. They were able to handle stress much better. No overt inflammation, again, immune status, healthy animal. This work was also published a couple of years ago. The last one that I want to cover, again, we're still, just to remind ourselves, we are still in stage 3 active immunomodulation, which is the work that was done by a colleague of mine, Gail, who many of you know about, on the SF68, the effect of probiotic SF68 on puppies. 14 puppies part of the study, 8 weeks of age, 2 diets, also a long 44-week trial. And the diets are listed here in terms of how they were supplemented: 5 into 10 raised to 8 CFU, enterococcal faecium, SF68 per day, and you had both gut immune and gut health measures monitored during the study. Here's the design of the study. Again, 8-week pre. You have the CDV vaccine, booster vaccine delivered, and then you start measuring over time. And what you see here is exactly the same pattern. You're looking at both systemic immune response shown here, both in terms of IgG as well as IgA. What we saw earlier was just looking at the overall titers, and clearly in both cases, kittens that were, or the puppies that was fed diet supplemented with the probiotic has significantly higher levels of response to CDV vaccine as compared to the control, both in terms of IgG or IgA, whichever one you choose to measure and interpret your response. You also had significant increase in the amount of fecal IgA as you start closer towards the end of the trial, which is what we saw as a pattern in others. Again, underlying the point that you need to give enough time for these nutritional interventions to have an impact on the immune system that could be measurable. So, bottom line, SF68 enhances systemic immunity as measured by the vaccine response, also enhances local immunity in terms of higher fecal IgA, which is a marker of a healthy and an active gut-associated lymphoid tissue. So that kind of summarizes a few examples that we talked about in terms of how can we actively modulate the immune system by engaging with the immune system with the ingredients that you choose to add as part of the diet. We've also done some studies with spirulina, which is blue-green algae, and we got very similar results. Again, I did not choose to put it because of the time constraints, but we can always talk about that if there's questions later. Now, stage 4 is where things get really interesting, where you're talking about personalizing immunonutrition for the need of the pet at that given time. And this is equally applicable to humans as well. There are several things, and I've just listed a couple to kind of get your thought processes going, but I'm hoping as some of the young researchers in the room, this list will go way beyond as we start diving and understanding how to engage with the immune system, not through drugs, but through nutrition. So examples are, can you induce a local Th2 response in the gut if there are situations where you have inflammatory bowel disease type syndrome? There are ingredients that are known to enhance TGF-beta, for example. Other ways for us to be able to have probiotics that can deliver anti-inflammatory cytokines locally. Now again, many of these things are doable from the science perspective, but again, me and many of our colleagues work in the interface between pure science and the applied component. And there's a lot that gets kind of lost in translation between science and application. And this is where, as we have more discussions here, as we talk about this science, many of these areas will kind of get elaborated and expanded, because there's also regulatory processes that we need to take in mind when you talk about probiotics. Because what I want to leave here is a flavor of what could be and what the future is likely to be. And I hope many of you in this room and others who are listening in would participate in really making this the reality that we can offer to both our pets as well as our family members, humans, through the work that we would do and some of you will engage with down the road. Again, all this amazing work that I talked about wouldn't have been possible with great colleagues. But anyway, I'll come back to the thank you part a little. But again, let's recap the discussions that we've had. Why enhance the immune health? We talked about reasons: malnutrition, life stage, stress, overnutrition, undernutrition could be some examples. And again, we're not touching about disease or fatigue induced by external factors like extreme exercise. How can we engage with the food, engage with the immune system using food is to be able to carefully choose ingredients that can impact key pathways. Some of these are known, some of these will be known down the road, and how can we come up with creative ways to be able to engage and interact and drive that response. And then finally, the framework that we talked about, stage 1, stage 2, passive, and you could argue about that, but you're really providing nutrients or enhancing levels of key nutrients. Stage 3 and 4, you're actively engaging with the immune system. Stage 3 is more irrespective of the individual, but stage 4 could be more personalized and specific. And again, all this work wouldn't have been possible with an amazing set of colleagues that I'm so fortunate to be able to work with. And I've listed here, Julie's right in the audience. She's been a great partner with me working on some of the microbiome work. I'm an immunologist. I did microbiology way back in my days. Let me stop there and open up for questions, comments that you might have. Thank you for your attention.